Well, are you ready to start now, Evan?
Sure, but how about a question? Let's see, what happens when you freeze my mouth?
Well, the local anesthetics are going to prevent any nerve impulses from traveling along the pain fibers that won't reach your brain, so you won't feel anything.
So, freezing stops my nerve impulses?
That's right. The nerves can't repolarize. Now, can you open?
But I just got another thing.
Come on, Evan, it's gonna be okay.
We give lots of people needles every day, and they all live to tell about it.
I'm just not every other person. Look at the size of that needle. I don't know which is
You know, by the end of this program, you and Evan are going to better understand the nature of the neural system and its role in maintaining equilibrium in humans. Different techniques will be used to illustrate neurons, and you're going to understand how chemicals can influence their functioning. By the end of the program, you're going to be able to explain the formation and the conduction of the action potential. We regulate our internal environments and physiology using electrochemical control systems. Neurons play an important role in this regulatory function. They communicate with each other and with the effectors of our bodies. If Evan ever gets an anesthetic in there, the impulses that are sent along sensory nerve fibers in his jaw will be affected. Let's see if you can remember what a neural pathway is.
Neurons are the structural and functional units of the nervous system. Within a neural pathway, a neuron is organized with axon terminals that almost touch other neurons at the dendrites, axons, or cell bodies. Impulses are conducted along a neuron from the dendrites to the axon and are transmitted between neurons from the axon of one neuron across the synapse to the dendrites of another neuron. Transmission of impulses between neurons involves neurotransmitters that span the synaptic cleft.
So, within a neural pathway, neurons can pass signals to each other, right?
That's right.
Okay, but how does the signal travel inside of a neuron?
Impulses travel through a neuron from its dendrites into the axon and along its length. Impulses can also travel from neuron to neuron, from the axon terminal of the presynaptic neuron to the postsynaptic neuron.
Okay. Say you were a neuron in a giraffe's leg. It would have to be pretty long, wouldn't it?
Yeah, it would. But the impulses would travel the same way through that neuron as it would, say, through the neurons in Evan's jaw. But before we get into how the impulse travels, we need to look at the electrophysiology.
So, Andrea, to record from these nerve cells, we use glass micropipettes. And this is glass capillary tubing, which we draw to a very fine tip. And then we press this glass tip against the nerve cell. We apply a small amount of suction to form a tight seal between the glass and the membrane. And then we apply a little bit more suction, and that breaks through the membrane and gives us a low resistance access to the inside of the cell. And this allows us to record activity of nerve cells while we're doing experiments.
How do you actually do this type of work when the cell is so tiny?
This is actually very tricky. It takes a lot of practice. You have a fine micromanipulator, and this allows you to make very small movements of the electrodes, and this controls 3 axes, and you have a hydraulic control of the manipulator, and the whole system is dampened from vibration, so there's no vibration going down there. One cell is extremely tiny, and so you use a manipulator to advance and push the electrode against the cell.
Do you have any pictures that you could show me of what the cell looks like?
Yes, we have a picture set up on the microscope here, and if you want to take a look, we can show you what a cell looks like under a microscope. So if you look through here, you can see the cell body is a large yellow fluorescent cell body. Defined processes are dendrites coming away from the cell body. This is where activity comes into the nerve cell. And the finer process, that's the axon, where the activity leaves the cell and goes out and then makes contacts with other cells.
Ion currents result from the movement of charged particles or ions across the neuron membrane. The ions diffuse through protein ion channels, dispersed widely throughout the membrane due to the establishment of concentration gradients. Passive diffusion occurs along the concentration gradient. Active transport works against the concentration gradient. Both passive diffusion and active transport occur at the same time. Active transport, and to a lesser degree diffusion, function to establish a slight positive charge outside the membrane and a slight negative charge inside. This is called resting membrane potential. Resting membrane potential is the electrical potential across the membrane when the neuron is not conducting a nerve impulse. It is about minus 70 millivolts. Resting membrane potential develops as a result of the movement of sodium and potassium ions across the cell membrane of the axon via the sodium-potassium pump. Some of the potassium ions that passively diffuse to the outside of the membrane are recaptured by the sodium-potassium pump. Active transport of ions leaves the axon polarized with positive charges on the outside and a net negative charge on the inside of the membrane. This is due to the uneven distribution of ions. More positive ions on the outside of the membrane than on the inside.
So this here represents the cell membrane, and this must be the ion channel, right?
That's right, and its activity is important for nerve impulse conduction. This plastic model represents just a segment of the axon.
Okay, so you've got more positive ions on the outside and negative ions on the inside. The purple and yellow ones represent the positive ions, and the white ones represent the negative ions.
That's right, and the difference in charge between the interior and the exterior is the membrane potential.
So negative inside and positive outside, that's polarity, right?
That's right. That means that this membrane is polarized. At rest, it's about negative 70 millivolts. In the intercellular fluid, the concentration of potassium ions is high and the concentration of sodium ions is low. The reverse is true for the extracellular fluid, where the sodium concentration is high and the potassium low.
Okay, Evan, you ready to go?
Okay, wait a second. Okay, if the sodium and potassium ions are positively charged, then how come the neurons in my jaw are polarized?
If Evan ever gets that needle, the neural membranes in his jaw will be affected. So, how are you going to show resting membrane potential?
Well, we found that the charge separation was due to the movement of the potassium and sodium ions.
Actually, we've created a simulation that'll show the movement of the sodium and potassium ions in and out of the cell. Okay, come over here, you guys. Mr. Leon, come back with me. Okay, I'll have you stand there.
Okay.
You guys line up over here and there. Okay, perfect. They're going to be the sodium ions in the yellow shirt, and we're going to be the potassium ions in the purple shirt. All right, I'll give you this, and you can be the sodium-potassium pump. This plastic is going to represent the cell membrane, and the slot here is going to be the ion channel.
Membrane potential is established as I actively transport the sodium ions from the inside of the cell through to the outside, and as I actively transport the potassium ions from the outside of the cell through to the inside. Active transport requires ATP, or adenosine triphosphate, for energy.
That's right. Now, in time, positively charged sodium ions build up on the outside of the cell, and potassium ions on the inside of the cell. Now, as the amount of potassium ions increase, a concentration gradient develops.
Now, the ion channels will only allow some of the potassium to diffuse through the cell membrane, out of the cell, leaving behind the negatively charged chloride ions.
Over time, the buildup of these ions results in a negatively charged interior. The membrane becomes polarized. The inside is negative compared to a positively charged exterior.
And that's how membrane potential is established.
Okay, Evan, here we go. This is it.
Oh, no. I know the part about the neuron. Resting membrane potential means polarization. But what about the nerve impulse?
Okay, Andrea, this is the manipulator I was talking about. This is what we use to position the microelectrode on the cell. The microelectrode you can see right here, that's attached to this holder, and this holder is held onto the micromanipulator here, and the suction is applied through this tubing that leads to the syringe, and then we use this syringe to apply suction onto the cell to actually record electrical activity from the cell.
So how do you actually record the electrical activity?
Well, to do that, we use all this equipment right here. This is all a set of amplifiers to record the activity, and then it's displayed on the screen here, showing what this actually a typical response from a nerve cell looks like. So we see here, this is a typical response from a nerve cell, where we see the resting potential of a minus 70 millivolts, and we have a small depolarization where the cells depolarize up to threshold minus 55 millivolts, which initiates an action potential, which is shown here. This is the action potential goes up and then comes back down, and is followed by an after hyperpolarization, after the repolarization of the action potential. And this is caused by a sudden increase of permeability to sodium in a nerve cell. And this is what's blocked by a lidocaine, a local anesthetic, when you go to the dentist.
Stimulation of the neuron causes a depolarization of the membrane. The stimulus causes a local increase in the membrane's permeability to sodium ions. Sodium enters the cell following the concentration gradient and is attracted to the negatively charged interior. This voltage change causes the sodium gates to open and positive ions to flood into the cell. When the sodium gates close, repolarization begins. The membrane becomes permeable to potassium, causing it to rush out of the cell. As the sodium-potassium pump moves sodium out and potassium back in, resting membrane potential is reestablished. Once depolarization occurs at one spot, an adjacent portion of the membrane is depolarized. This wave of depolarization and repolarization, or action potential, travels along the axon. This is how the nervous impulse is propagated. A refractory period or short interval occurs after an action potential. During this time, the neuron cannot respond to another stimulus until it is repolarized. Nerve impulses travel between 0.5 to 120 meters per second. Refractory periods last 1 to 2 milliseconds. That means a nerve can carry impulses at a frequency of 1,000 times a second. This rapid communication in the neural system helps the human organism maintain control over its internal environment.
Okay, that's great. Good. Doing fine. Just about done. We got it. Okay, ready.
Well, now that I got the Novocaine, could you run that polarization and repolarization stuff again?
Yeah, sure. Let's start right at the beginning.
All right, ions, establish resting potential. All right, you ions, establish resting potential. And prepare for stimulation and depolarization wave. Prepare for stimulation and depolarization wave. All right, ready? 3, 2, 1. Stimulation.
Stimulation of the neuron causes a depolarization of the membrane. Depolarization involves the movement of sodium ions. The positive ions rush in, leaving the outside negatively charged. Immediately following, the potassium channels open and potassium rushes out, leaving the membrane to repolarize. Meanwhile, the adjacent membrane is depolarized. A brief refractory period follows where the neuron cannot be depolarized.
Well, how are you feeling there, Evan?
Okay.
Good. Are you starting to get numb in this area yet?
Yeah.
All right.
So, what gets the wave of depolarization, repolarization going?
The wave of depolarization involves the all or none principle. Let's see if you can figure it out. All right, what did you observe?
Well, these two rows collapsed, but this one didn't.
Yeah, with this row, it didn't even look like you gave it a strong enough push for them to move.
Okay. What about rows A and B?
Well, I think row A got a stronger push than row B, but they still both collapsed.
That's right. And that's the all or none principle. Stimulus must be of sufficient intensity to cause depolarization. It doesn't matter if the stimulus is stronger. Row C received a stimulus that wasn't strong enough. It didn't reach threshold potential. So stimulus was below threshold, a subthreshold stimulus.
So the threshold potential wasn't reached, and therefore depolarization couldn't occur.
The push that the dominoes received wasn't strong enough to move them.
That's right. Let's take a look at this another way.
What do you mean?
Oh, watch. A wave illustrates the all or none response. Threshold level is required to get the wave to start. A stimulus, a people, the moment. Once started, the wave will propagate around the stands.
Yeah, but we can see it here. In a neuron, it must move a whole lot quicker.
Just how fast does that impulse travel?
All right, Evan, can you open wide and try not to move?
Are you okay, Evan?
Uh-huh.
It only takes milliseconds for the nerve impulse to travel along the axon. A stimulus is received, passed along sensory nerves, to the central nervous system, and then to motor nerves and effectors. This impulse is actually the series of action potentials propagated along the length of the neuron. The rate of conduction of the impulse is affected by two factors: the diameter of the axon and the myelin sheath. The greater the axon diameter, the faster the rate of conduction. Some invertebrates, like squid and lobster, have giant axons which conduct impulses rapidly. Vertebrates have supporting cells which form a thick myelin sheath around the axons. Schwann cells grow and wrap their membranes around the axon. This forms concentric layers. There are small gaps in the fatty sheath called nodes around the A, where the Schwann cells join. Voltage-sensitive ion channels are concentrated in the node regions. Action potentials can only be generated at these nodes, causing the nerve impulse to jump. The impulse jumps from node to node. This results in a faster rate of transmission called saltatory conduction. The difference in conduction between myelinated and non-myelinated neurons can be illustrated in this next simulation. In myelinated neurons, the membrane is polarized primarily at the nodes between the Schwann cells. The distance between these students is greater, representing the nodes in the myelinated neuron. These students will represent the non-myelinated neuron. The wave of polarization will travel faster down the myelinated neuron as it jumps from node to node. These different neurons are located in different locations in the human nervous system. We have specialized neural tissues to help us maintain internal control and maintain equilibrium with the external environment. Substance and chemical abuse has a sociological and biological basis. Studies show that ethanol affects neural membrane proteins. The precise mechanism is unclear. If these neurons are in the central nervous system, behavior and physiology are altered. Changing cellular processes affects the tissues of biological systems and equilibrium. The electrochemical nature of the nerve impulse is part of a diagnostic tool. Here in the EMG lab, neurologists perform electromyographic examination of neural and muscular systems. Let's check in on Dr. Mary Ann Lee and see her doing one of these EMGs.
Welcome again.
Hi, Dr.
You're
Lee.
here for our demonstration.
Yes, we are. At
Come on
last.
over.
Okay. Thanks. We have a patient ready, or?
We have a volunteer patient. Jane's agreed to let us demonstrate how we do a thing called nerve conduction studies.
Excellent.
Now, we refer to this as EMG. We always talk about patients coming for an EMG, which stands for electromyography. But that's not totally true. That's only part of what we do in this lab. The other part is what I just mentioned, which is called nerve conduction studies. And this is looking at the function of the peripheral nerves. The EMG part is looking at the electrical activity within muscle, and that involves putting a needle electrode into the muscle. We're not going to be doing that today. We do these tests for various different problems that can affect either the peripheral nervous system or muscles. What I'm going to demonstrate today is part of the testing we do for a very common peripheral nervous system problem called carpal tunnel syndrome. What we're going to do is, first of all, look at what we call sensory responses. That means we're going to stimulate the sensory fibers in the nerve and record the response from higher up. We put our surface electrodes that are going to record the response further up at the wrist, and we use a set distance, which is 13 centimeters. To move this a bit. Now we put these surface electrodes on, and that one's the reference electrode. And this is the active electrode, and this is over the median nerve, which is right down in the middle of the wrist. Our fancy machine here, we start with a very small electrical stimulus of about 1 or 2 milliamps. And that'll get a bit stronger, and we keep increasing the stimulus until the response doesn't grow any further. And that usually is somewhere around 20 milliamps. We're getting a nice sensory nerve action potential here. Jane, you have a lovely response.
Good.
And the conduction velocity, or the speed that the electrical impulse is traveling in that nerve, is 55.7 meters per second. So that's pretty fast. And that's normal.
That's normal.
That's all normal. So that was the sensory response. Now what we'll do is we'll go over, we'll look at the motor response. And for that we change electrodes. With the motor response, we go the opposite direction. We stimulate the nerve and we record from the muscle. So we're recording a totally different response this time, and it's called a compound muscle action potential. And we're going to be stimulating in 2 different locations. First will be here at the wrist, and the carpal tunnel is in this area here. So obviously this stimulus is going to measure conduction through the area that we're interested in. But in also to get conduction velocity, we stimulate in a second place, and in this case we'll do it up at the elbow so that we can measure what the normal conduction velocity is in the forearm here. And I just have to change my stimulating electrode here as well. Okay, now Jane, with this one, it's going to feel very much like what you felt in your finger, except this one will build up and it's going to make your thumb jump. Okay? There it is.
I saw a thumb jump.
That's right. What's that feel like?
It was a bit stronger than on the finger.
Okay, here we go again. And like we did on the finger, we increase the intensity of the stimulus until the response doesn't get any larger. Now we're going to stimulate the median nerve at the elbow, and we know our anatomy well enough to know roughly where this nerve is supposed to be. And this is going to make not only your thumb but a bit of your arm jump as well. And there we've got our response. And we mark where we stimulated up here.
So it's great motor nerves.
It's got Good motor nerves and good sensory nerves and no carpal tunnel.
Electrical nerve stimulation can also be used as an alternative to local anesthetic. Let's see how this technology works.
Okay, Evan, I'm going to get Paula to attach some electrodes and we'll show you our TENS machine. Okay. Get you to hold that.
What?
Now, TENS stands for Transcutaneous Electrical Nerve Stimulation. And when you turn this on, the electrical impulses will activate inhibitory interneurons. And this will prevent any signal from a pain fiber from reaching your brain, so you won't feel anything. So I'm going to get you to turn it on.
Okay.
And just turn it up until you start to feel some tingling, and then just leave it there. At that point.
Okay. Where can I feel it?
Good. Unfortunately, it only works in about 50 to 70% of the population, so we'll have to see how effective it is for you. I'll try turning up a bit higher now.
Okay. It's too bad that Evan is part of the population that has to rely on more traditional anesthetic. How an anesthetic works isn't entirely clear, but it is believed to impair the ion permeability of the axon. That means that the membrane can't depolarize or repolarize again. Therefore, no message, no impulse, no pain.
Medical records show that from 10 to 25% of the general population suffers from migraines or periodic debilitating headaches. Up until recently, it was thought that the neurotransmitter serotonin was somehow responsible for the constriction of cerebral blood vessels, followed by dilation of the same vessels in the brain. It is now recognized that migraines are complex neurological disorders. Neurobiologists have proposed that migraines are initiated by a massive depolarization or excitation of neurons, known as spreading depression, in the subregions of the brain cortex. This wave of depolarization stimulates the peripheral terminals of C nerve fibers that innervate the blood vessels on the surface of the brain. C nerve fibers are very small in diameter and conduct impulses at a rate of about 1 meter per second. Stimulation of the C fibers results in the vascular component of the headache and the accompanying pain.
Oh, hey, how'd it go?
Oh, nice lift.
Thanks. It went okay.
Looks like you sure got the message about anesthetics. Too bad the TENS machine didn't work for you.
Hey, I guess you really learned that the neural membrane is polarized because of the transport of sodium and potassium ions.
Okay, whatever.
And that the nerve impulse is the movement of action potential along the axon.
Sure.
Depolarization and repolarization is when the resting potential is reestablished.
Like the wave.
Oh, okay, the wave. Yeah.
Yeah. But you have to be sure that it's a strong enough stimulus because it is an all or none response.
Woohoo!